Good day, dear students. I am Chimidonov Sergei Nikolaevich, the head of Human Anatomy Department in Samara State Medical University. Today we'll talk about anatomy of brain stem. The brain encephalon occupies the cavity of the skull, and its shape corresponds in outline to the inner contours of the cranial cavity. It has supralateral or dorsal surface. It also convex in conformity with the cave area, whereas the ventral surface of the base of the skull is more or less flat and uneven. Three large parts are distinguished in the brain, namely the cerebral hemispheres. You may see this part here, cerebellum, and the brain stem, or truncus cerebri. So you may see these parts here. You may see the cerebral hemispheres. You may see right hemisphere. Also you may see the half of cerebellum, and also you can see the stem. The brain organization you may see here. It includes the brain stem. Consists from four parts: medulla oblongata, pons, mesencephalon, diencephalon. And also the brain contains cerebellum and telencephalon with two cerebral hemispheres. Let's talk about development of the brain. At the early stage of the development, the neural tube is subdivided in two parts corresponding to the brain and the spinal cord. The anterior expanded part, the rudiment of the brain, is divided by constrictions in the three primary cerebral vesicles situated one next to the other: anterior or forebrain, or prosencephalon, it's in Latin, middle part or midbrain, and also we talk in Latin mesencephalon, and posterior or hindbrain or rhombocephalon. So you may see these three parts here on this picture. This is prosencephalon or forebrain. In red color you may see mesencephalon, it's in Latin, or midbrain in English, and in pink color you may see rhombocephalon or hindbrain. So the anterior vesicle is closed in front by a lamina terminalis. This stage of three vesicles transformed during subsequent differentiation into five vesicles. And you may see five vesicles here, which give rise to the five main parts of the brain. At the same time, the neural tube curves sagittally. You may see here. A dorsally convex cephalic flexure forms first in the region of the midbrain, and then another dorsally convex cervical flexure appears at the junction with the spinal cord rudiment. Between them, in the region of the hindbrain, forms a third ventrally convex curvature, the Pantin flexure. You may see the Pantin flexure here. The last named curvature divides the posterior brain vesicle, the rhombencephalon, and you may see a rhombencephalon here. In two portions, the posterior portion in number 5. The myoencephalon transfers finally into the medulla oblongata, while the anterior portion in number 4, you may see it here, develops into the pons or Verolli pons. Here, look at my pointer, and cerebellum in number 4 in dorsal side. A narrow constriction, the isthmus rhombencephaly. You may see in yellow color here between a rhombencephalon and a mesencephalon, between hind brain and midbrain in yellow color. So the isthmus rhombencephaly separates the metencephalon from midbrain. The common cavity of the rhombencephalon, a rhomboid-shaped on frontal section, forms the fourth ventricle, communicated with the central canal of spinal cord. Its ventral and lateral walls are considerably thickened due to the development of the nuclei of the cranial nerves in them, while the dorsal wall is thin. The greater part of this dorsal wall in the region of the medulla oblongata consists only of a single epithelial layer which fuses with the pia mater, tela chorioidea inferior. The walls of the mesencephalon thicken more uniformly with the development of the medullary matter in them. Ventrally, they give rise to the cerebral peduncles, dorsally to the tactile lamina. The cavity of the mesencephalon is converted in narrow canal, the aqueduct. You may see the mesencephalon in red color here. So the aqueduct communicating with the fourth ventricle. The differentiation and change in shape of the prosencephalon are more marked. It is subdivided into a posterior part, diencephalon. You may see number 2 in blue color. And anterior part, the telencephalon. You may see in brown yellow color here in number 1, telencephalon. The lateral walls of the diencephalon thicken to form the thalami. Besides lateral evaginations of these walls form 2 optic vesicles. You may see optic vesicles here, from which ultimately develop the retina and the optic nerves. The dorsal wall of the diencephalon remains thin in the form of an epithelial plate which fuses with the pia mater. A protrusion of this wall forms posteriorly, from which the pineal body, or corpus pineale in Latin, is derived. The hollow peduncles of the optic vesicles are drawn on the ventral side into the wall of the prosencephalon, as the result of which a depression, the optic recessus opticus, forms on its floor. The anterior wall of recess is formed by the thin lamina terminalis. To the back of the optic recess, another funnel-shaped depression forms its walls, give rise to the tuber cinereum, the infundibulum, and the posterior neural lobe of hypophysis cerebri. Still farther to the back, in the region of the diencephalon, the paired mammillary bodies, corporomobilaria, are laid down. The cavity of the diencephalon forms the third ventricle. I will show you these parts of diencephalon later. So let's talk about development of telencephalon. Let's talk some words about this. The telencephalon is divided into a smaller medium part, telencephalon medium, and 2 large lateral parts, the vesicles of the cerebral hemispheres, hemisferum dextrum and sinistrum, which grow intensively in man, and when fully developed are much larger than all the parts of brain. The cavity of the median telencephalon being a continuation of the cavity of the diencephalon, the third ventricle, communicates on each side with the cavities of the hemispheric vesicles by means of interventricular apertures. So you may see these apertures here, and the third ventricle communicates with lateral ventricles. So the anterior wall of the median telencephalon is a continuation of the lamina terminalis. At the beginning of the first month of embryonic life, it forms a thickening coat the chamisaral lamina, from which the corpus callosum and the anterior chamisure develop eventually. So you may see this part of cerebrum here. This is corpus callosum. A projection forms in the base of the hemispheric process on the inner surface on either side. This is nodular tuber from which the corpus striatum develops. Part of the medial wall, so you may see the corpus striatum here, this one. So part of the medial wall of the hemispheric vesicle remains in the form of a single epithelial layer invaginated into the vesicle as a fold of the pia mater, plexus chorioideus. You may see this plexus here. In diverticulum, the rudiment of the rhinocephalon forms on the inferior surface of each hemispherical vesicle as early as the 5th week of embryonic life. The diverticulum is gradually delimited from the wall of the vesicle by a fissure which corresponds to the fissure renalis laterally. As the gray matter cortex and then white matter develop in the walls of the hemispheric, the latter grows and forms a mental pallium, situated above the rhinocephalon and covering not only the thalami but also the dorsal surface of the midbrain and the cerebellum. So you may see this gray matter here, cortex cerebri. The growing hemisphere increases in size first in the region of the frontal lobe. So you may see also frontal lobes here and here. Then the parietal and the occipital lobes are, and finally in the region of the temporal lobe. As a consequence, it seems that the pallium rotates as it were about the thalami first, from front to the back, then downward, and finally course over forward to the frontal lobe. As a result, the lateral cerebral or sylvian fossa, you may see this fossa here, fossa cerebri lateralis, forms on the lateral surface of the hemisphere between the frontal lobe and the temporal lobe, which has moved closer to it. When these two lobes come into intimate contact, this fossa transforms into the lateral cerebral or sylvian sulcus, sulcus cerebri lateralis. You may see this sulcus here. And in details, we will talk about this sulcus when we will study telencephalon. With the development and growth of the hemisphere, its internal cavities, lateral ventricles of brain, as well as part of corpus striatum, develop together with it and go through the mentioned rotation. This explains the similarity of their shape to that of the hemisphere, the presence of an anterior, central, posterior and inferior parts in the ventricles. The human brain grows intensively in the embryonic period and during the first years of life, when the organism grows vigorously, becomes adapted to the new environment and acquires an erect posture, and when the second verbal signaling system is formed. The growth of the brain is completed by the age of 20 years old. The brain of newborn weighs 340 grams in boys and 330 grams in girls. The brains of an adult male weighs 1 kilogram and 375 grams, and adult female 1,245 grams. So let's talk about parts of brain. So I told you about vesicles of brain. You may see a rhombencephalon in pink color, number 3rd. Rhombencephalon or hindbrain consisting in turn the myoencephalon. You may see number 5 myoencephalon here, or medulla oblongata, it's in Latin, and metencephalon, hindbrain proper. And we talk about develops of cerebellum and pons. The mesencephalon in red color in number 3, or midbrain, and also prosencephalon or the forebrain, in which are distinguished the diencephalon in blue color in number 2 and telencephalon. So diencephalon in English is between brain, or telencephalon is and brain. It's in English. All the parts named with the exception of the cerebellum and telencephalon form the brain stem. Brain stem. You may hear these parts again, brain stem. Besides this part, the isthmus rhombencephaly, as I told you, lying between the rhombencephalon and mesencephalon. So the mesencephalon and the prosencephalon constitute the cerebrum as distinct from the cerebellum. So first we'll talk about hindbrain, and we'll talk about medulla oblongata. Medulla oblongata or myoencephalon is a direct continuation of the spinal cord into the brain stem and is part of the rhombencephalon. So you may see the part of spinal cord, and also you may see the myoencephalon or medulla oblongata here. It combines the structural properties of the spinal cord and the initial part of the brain, hence the name myoencephalon. In Greek language, myos narrow, encephalos is brain. The medulla is shaped like a bulb, like onion or garlic. You may see in details here. So it's in Latin, this is bulb. And the second name of this part of brain is bulbar part. Bulbus cerebri, it's in Latin. So its upper expanded end borders upon the pons. You may see here. And its inferior boundary is the site of emergence of the roots of the first pair of cervical nerves at the level of the foramen magnum of occipital bone. So, on the midline of the anterior ventral surface of the medulla stretches the anterior median fissure, which is a continuation of the anterior median fissure of the spinal cord. You may see here. On the either side of its longitudinal elevation called the pyramids of the medulla oblongata. Pyramides, medulla oblongata, it's Latin. Both pyramids and continues with the anterior funiculi of the spinal cord. Some of the bundles of nerve fibers composing the pyramid decussate, you may see the decussate here, in the depth of the anterior median fissure with the bundles of the contralateral pyramid to form the decussation of the pyramids and then descend in the lateral funiculus of the contralateral side of the spinal cord. This is the lateral corticospinal or pyramidal tract, tractus corticospinalis in Latin. Those bundles which do not descend in the anterior funiculus on their own side and form the anterior corticospinal or pyramidal tract. The pyramids are absent in the lower vertebrates. They appear with the gradual development of the neurocortex and are therefore most developed in men because the pyramidal fibers connect the cerebral cortex, which attains the highest development in men, with the nuclei of cranial nerves and with the anterior horns of the spinal cord. Lateral to the pyramid is an oval swelling, you may see here, aliva, separated from it by fissure anterior lateral sulcus or sulcus. On the posterior dorsal surface of medulla is a posterior median fissure, sulcus medianus posterior, a continuation of the posterior median fissure of the spinal cord. Lateral to it are the posterior funiculi bounded laterally. You may see part of this fissure here. Above the posterior funiculi diverge laterally and pass to the cerebellum to be components of its inferior pedicles, pedunculis cerebellaris inferioris, bounding the rhomboid fossa below. You may see the part of inferior pedunculis here. So, at the inferior angle of the rhomboid fossa, both funiculi acquire the elevation known as a tuberculum nuclei gracilis and tuberculum nuclei cuneati. You may see in schematic drawing here tuberculum nuclei gracili, tuberculum nuclei cuneati. These elevations are formed by gray matter nuclei, called the gracile nucleus and cuneate nucleus. The ascending fibers of the posterior roots of the spinal cord, which pass in the posterior funiculi, terminate in these nuclei, fasciculi of Gowley and Burdach. The lateral surface of the medulla oblongata between the anterolateral and posterolateral fissures corresponds to the lateral funiculus. The 11th, 10th, and 9th, you may see here, 11th, 12th, 10th, and 9th pairs of cranial nerves emerge from the posterolateral sulcus behind the alive. You may see these nerves here. This one is 11th accessory nerve. You may see vagal nerve. It's 10th pair, and you may see glossopharyngeal nerve, 9th pair, and they arise from dorsolateral sulcus. So, the internal structure of medulla oblongata. The medulla oblongata originated in association with the development of the organs of statics and acoustics and the brachial apparatus concerned with respiration and circulation. That's why nuclei of gray matter related to the balancing and coordination of movements and to the control of metabolism are embodied in it. The olivary nucleus, you may see here, olivary nucleus, has the appearance are a carrot lamina of gray matter, open medially. This is hilus. It causes the external building of the alveoli. The alveolar nucleus is connected with the cerebellodentate nucleus and is the intermediate nucleus of balance, which is most pronounced in man whose erect posture needs the most perfect apparatus of balance. The reticular formation, you may see this part here, is formed of interlaced nerve fibers and nerve cells lying between them. The reticular formation of medulla oblongata is connected with the spinal cord. A bundle of descending fibers, the reticulospinal tract, passes from it to the spinal cord. The nuclei of the lower four parts of the cranial nerves, 12th to 9th, concerned with the narration of the derivatives of the branchial apparatus and the viscera. The vitally important respiratory and circulatory centers connected with the nuclei of vagus nerve. Injury of the medulla oblongata may therefore cause to death. The white matter of the medulla oblongata contains long and short tracts. The long tracts are as follows. The descending pyramidal tracts passing by transit into the anterior funiculi of the spinal cord and partly decussating in the region of the pyramids. You may see pyramids here, decussatio pyramidum here, and also you may see white matter. In addition, from the nuclei of the posterior funiculi, the grasso and cuneate nuclei arise the second neurons of the ascending sensory tracts passing from the medulla oblongata to the thalamus. These form the bulbothalamic tract, tractus bulbothalamicus. The fibers of this bundle form a medial loop, the medial limniscus, with which decussates in the medulla to form the decussatio limniscorum. So you may see this decussatio here. Thus, in the medulla oblongata, there are two decussations of the long conduction tracts, namely a ventral or motor decussation, you may see it here, or pyramidal decussation, and dorsal decussation of limniscus, dorsal sensory decussation of limniscus. The short tracts include the bundles of nerve fibers connecting some nuclei of gray matter to one another, as well as bundles connecting the nuclei of the medulla oblongata with the adjacent parts of the brain. Among these, the aliva cerebellar tract and the medial longitudinal bundle, fasciculus longitudinalis medialis, stretching dorsal to the inter-olivary layer deserve mention. The topographic relation of the principal structures of the medulla oblongata are seen on transverse section made through the olives. You may see here. The roots arising from the nuclei of the hypoglossal and vagus nerves divide the medulla on either side into three regions: posterior, lateral, and anterior. Posterior region contains the nuclei of the posterior funiculus and the inferior cerebellar peduncle. The lateral region contains the alveolar nucleus and the reticular formation, and the anterior region contains the pyramids. So you may see the 12th pair of nerve here, hypoglossal nerve, and also you may see vagus nerve and crossing section on its level. Next, we will talk about metencephalon. Metencephalon consists of two parts: ventral part, the pons, you may see it here, and also you may see it here, pons, and dorsal part of the cerebellum. The pons. The pons is seen on ventral part of brain, is a thick white rounded ridge bordering caudally upon the superior end of the medulla oblongata and cranially upon the cerebropeduncles. The lateral boundary of the pons is a line drawn through the roots of trigeminal nerve. You may see the trigeminal nerve here in number 5th. And facial nerve. You may see number 7th. So, this drawn line is the border between pons and cerebellum. Lateral of this line you may see the middle peduncles of cerebellum. So the dorsal surface of the pons is not visible because it's hidden by the cerebellum. It forms the upper part of the rhomboid fossa, the floor of the fourth ventricle. You may see this part here, and also you may see the middle peduncles of cerebelli. The ventral surface of the pons is of a fibrous character, with the fibers passing transversely, you may see here, for the most part and the direction of the middle cerebellar peduncles. A shallow basilar sulcus, you may see it here, sulcus basilaris, lodging the basilar artery, stretched on the midline of the ventral surface. So, if we look at the internal structure of the pons, you may see this transverse section, and you may see that the pons consist of a larger lower or ventral part here and smaller dorsal part. The boundary between them is formed by a thick layer of transverse fibers, the trapezoid body, corpus trapezoidum, or the superior salivary nucleus, also related to the auditory tract, is situated in the region of the trapezoid body. The nucleus is given the latter name because it has a dentate shape similar to that of the salivary nucleus in medulla oblongata. Pars ventralis contains longitudinal and transverse fibers. You may see longitudinal and transverse fibers here. Longitudinal fibers belong to the pyramidal tracts, the cerebropontine fibers, fibra cortica pontine, which are connected to the pontine nuclei, from which arise transverse fibers passing through the cerebellar cortex, the pontocerebellar tract, tractus pontocerebellaris. You may see these transverse fibers here, and that passes into cerebellar peduncle. So this house system of conduction tracts links the cortex of the cerebral hemispheres with the cortex of the cerebellar hemispheres through the pons. The more is the cerebral cortex developed, the more are developed the pons and cerebellum. The pons is naturally most pronounced in man, which is a specific feature in the structure of his brain. Pars doxalis contains the reticular formation of the pons, which is a continuation of the similar part of medulla oblongata. Above reticular formation is the floor of the rhomboid fossa, lined with the ependyma. Below the floor lie the nuclei of the cranial nerves, 8 to 5 pairs. The conduction tracts of the medulla oblongata also pass into the pars dorsalis and stretch between the midline of the dorsal nucleus of the corpus trapezoidum. They are component of the medial lemniscus, in which the ascending tracts of the medulla oblongata. You may see medial lemniscus here. So you may see a rhomboid fossa here, and anterior part or superior anterior part is the dorsal surface of pons, and posterior part or posterior inferior part is a dorsal surface of medulla oblongata. And also you may see the nuclei of cranial nerves here. You may see the cranial nuclei, trigeminal nerve in blue color. This is 5th pair. You may see 8th pair of vestibulocochlear nuclei, and also you may see 6th pair, the nucleus of 6th pair, nucleus motorius of abducens nerve, abduct nerve, and also you may see nucleus of facial nerve here. All these nuclei are situated in pons, and also you may see the nuclei of medulla oblongata. So if we talk about nerves, you may see all these nerves here, and also you may see the name of their nerves. So nu pair is nervus terminalis. First pair are nervi olfactorii. Maybe 18, 20 nerves here, nervi olfactorii. Nervus opticus is the second pair, and all these pairs have no nuclei, and also they are sensoric nerves. And you may see other pairs. Neurus oculomotorius contains parasympathetic and muscular fibers, or motor fibers, not muscular, motor fibers. Trochlearis nerve, it contains only motor fibers to muscle, skeletal muscle. Nervus trigeminus, it contains sensoric and motor fibers. 6th pair is nervus abducens, it contains only motor fibers. 7th pair, it contains 2 nerves, facial nerve and intermedius nerve. Facial nerve contains only motor fibers, and intermedius nerve contains sensoric and parasympathetic fibers. Also you may see 8th pair, nervus vestibulocochlearis. It contains 6 pairs nuclei. 2 nuclei are cochlearic, and 4 nuclei are vestibularic nuclei, and all of them are sensitive. 9th pair is nervus glossopharyngeal. It contains 3 types of nuclei and fibers: motor fibers, sensoric fibers, and parasympathetic fibers. And also all these fibers contains nervus vagus in number 10. The 11th pair is nervus accessorius. It's only motor fibers contained, and you may see 12th pair. This is nervus hypoglossus. It contains only motor fibers. So let's talk about cerebellum. The cerebellum is derived of the hindbrain, which developed in connection with the receptors of statics. It's therefore concerned directly with the coordination of movements and is an organ of the body adaptation to overcoming the main properties of its mass, gravity and inertia. It's also considered one of the highest centers of the vegetative sympathetic nervous system. The development of the cerebellum passed through the three main stages in process of phylogenesis and accordance with the change in the mode of the animal's movement. In the first stage, when the animals moved in water, you may see fish brain, by means of snake-like movements. The cerebellum had the appearance of the small structure, the auricula, which the terrestrial animals transformed into the flocculus. This is the oldest part of the cerebellum, or archicerebellum. In the second stage, when the aquatic animals acquired fins and the body movements changed, the new part of cerebellum forms the vermis. So you may see the vermis here, which in man is an outer or of the cerebellum or paleocerebellum. Finally, in the third stage, the cerebellar hemispheres appeared in the terrestrial animals. These are the newest part of the cerebellum or neocerebellum. They gradually grew as the animal's body was drawn farther away from the ground and movement by means of limbs developed. The cerebellar hemispheres attained highest development in man due to the movement in erect posture by means of one pair of limbs, the lower limbs, and the perfection of the grasping hand movements in labor processes. The cerebellum is situated under the frontal lobes of the cerebral hemispheres, dorsal to the pons and medulla oblongata, and is lodged in the posterior cranial fossa. The large lateral parts, the hemispheric cerebelli, and a narrow middle part lying between them, the vermis, are distinguished in it. On the anterior border of the cerebellum is the anterior notch embracing the adjoining part of the brain stem. The posterior border has narrower posterior notch separating the hemispheres one from the other. The surface of the cerebellum is covered with a layer of gray matter constituting its cortex and forms narrow cerebellar folia of the cerebellum, separated one from another by fissures. You may see these fissures and folia cerebelli here and also here. The horizontal and other large fissures divide the entire surface of the cerebellum in two lobes, lobuli cerebelli. Among these, it's necessary to point out the most isolated small lobule, the flocculus. You may see the flocculus here. Flocculus. Lying on the inferior surface of each hemisphere near the middle cerebellar pedicle and the nudulus. You may also see nudulus here, a part of vermis connected with the flocculus. The flocculus is joined to the nodulus by means of a fine strand, the peduncle of flocculus, which is continuous medially with a thin crenate lamina, the inferior medullary velum. Paired nuclei of gray matter are embedded in the white matter of both halves of the cerebellum. On either side of the midline, where the vestigium projects into the cerebellum, is the extreme middle nucleus fascigite. You may see the nucleus fascigite here. So, lateral to it lie small islets of the nucleus globosus. You may see here nucleus globosus. And still further lateral, the embaliforms nucleus. Finally, in the center of the hemisphere is the dentate nucleus, which has the appearance of the great heart lamina, resembled the alveolar nucleus, and has a mouth open medially, or the hilus or nucleus dentatus. The resemblance of the cerebellar dentate nucleus to the crenate nucleus of the alive is not a chance phenomenon. Both nuclei are connected with the conduction tracts, the fibra oliva cerebellaris. To each volume of one nucleus is similar to the volume of the other nucleus. Thus, both nuclei contribute together to the accomplishment of balance. The listed cerebellar nuclei differ in the phylogenesis age. The nucleus fascigui is related to the outermost part of the cerebellum. The flocculus archicerebellum, which is connected with the vestibular apparatus, and also I repeat that this is nucleus fascigui. The nuclei embaliformis and globosus here are constituents of the out part or paleocerebellum, which originated in association with the movements of the body. The nucleus dentatus here is related to the youngest part of the cerebellum, neocerebellum, which developed in association with the movements of means of limbs. Therefore, damage of each of these parts causes damage to different forms of motor activity, corresponding to the different stage of phylogenesis. Body balance is impaired in damage of the floccular nodular system and its nucleus fascigui, damage to the vermis and the nuclei embaliformis and globosus, corresponding to it leads to disturbed activity of the neck and trunk muscles. Damage to the hemispheres and dentate nucleus results in impaired activity of the limb muscles. The white matter of the cerebellum has on section the appearance of small leaves, you may see here, of a plant which correspond to each volume covered on the periphery by a cortex of gray matter. As a result, the general pattern of the white and gray matter on section resembles a tree, and it's called arbor vitae cerebelli. This names the tree of life, tree of life or arbor vitae, you may see here. So the white matter of the cerebellum is composed of different types of nerve fibers. Some of them link the folia and lobules, others pass from the cortex to the nuclei embedded within the cerebellum, still others connect the cerebellum with the adjoining parts of the brain. The last mentioned fibers are components of the three pairs of cerebellar pedicles. So you may see cerebellar pedicles here. This small part are superior, the great part is middle part or middle cerebellar pedicles, and the posterior cerebellar pedicles. The inferior cerebellar pedicles. The inferior cerebellar pedicles, pedunculis cerebellaris inferiores, running to the medulla oblongata, convey to the cerebellum the tractus spinocerebellar or flexic tract or posterior spinocerebellar tract. The fiber acquired externa from the nuclei of the posterior funiculi of the medulla oblongata and the fibra olivocerebellaris from the ulna. All these fibers and in the cortex of the vermis and hemispheres. In addition, fibers pass in these peduncles from the nuclei of the vestibular nerve and end in the nucleus fasciae. These fibers convey to the cerebellum impulses from the vestibular apparatus and the proprioceptive field. As a result, it becomes a nucleus of proprioceptive sensation concerned with automatic correction of the motor activity of the other parts of the brain. Descending tracts also pass in the opposite direction and the inferior peduncles. From the nucleus fasciae to the lateral vestibular nucleus and from this nucleus to the anterior horns of the spinal cord, the vestibulospinal tract. The cerebellum produces an effect of the spinal cord through this tract. The middle cerebellar peduncles contain fibers passing from pontine nuclei to the cerebellar cortex. The pontocerebellar tracts, the conducting pathways to the cerebellar cortex, arise in the pontine nuclei and are a continuation of the corticopontine fibers which terminate in the nuclei of the pons after decussation. These pathways link the cerebral cortex with the cerebellar cortex, which explains the fact that the more developed in the cortex of brain, the more developed are the pons and the cerebellar hemispheres. This is encountered in man. So the superior peduncles here, a little part you may see here and also here on this picture. So the superior peduncles, pedunculi cerebellaris superioris, it's in Latin, running to the tactile lamina of mesencephalon or midbrain, consist of nerve fibers stretching in both directions. To the cerebellum first, the anterior cerebellar or converse tract or tractus spinocerebellaris anterior, and from the cerebellar dentate nucleus to the tectum, the cerebellar tegmental tract, which after decussation terminates in the red nucleus of and thalamus. The cerebellum receives impulses from the spinal cord through the first tract and sends impulses to the extrapyramidal system along the second tract, and thus it itself has influence on the spinal cord through this system. So next, we talk about isthmus rhomencephaly. You may see some parts of isthmus rhomencephaly here and also here. Isthmus rhomencephaly is the junction of the rhomencephalon with the mesencephalon. Its components are as follows: the superior cerebellar peduncles. You may see these peduncles here. The superior medullary velum here, it's cutted. A superior medullary velum stretched between these peduncles, anterior or superior peduncles of the cerebellum, and also trigonum lemnisci, formed by the auditory fibers passing to the lateral lemniscus. The gray-colored triangle is bundled by the inferior quadrigemal brachium anteriorly, you may see here, by the superior cerebellar peduncle posteriorly here, and cerebral peduncle laterally. Here, this one. The last named is separated from the isthmus and mesencephalon by clearly defined sulcus lateralis mesencephaly. The upper end of the first ventricle continues with the aqueduct in the mesencephalon, projects into the isthmus. Next, we talk about fourth ventricle. You may see here, and also you may see here, fourth ventricle is a remnant of the cavity of the posterior cervical vesicle and is therefore the common cavity of all parts of hindbrain, composing the rhombencephalon, a tent in which a floor and root are distinguished. So let's look at sagittal section. You may see sagittal section, and also you may see fourth ventricle. You may see the tent of fourth ventricle and or root, and you may see the floor or a rhomboid fossa. And also you may see velum medullaris superius and inferius. So the floor or base of this ventricle is shaped like a rhombus, I told you, which seems to be pressed into the posterior surface of the medulla and pons. It's termed the rhomboid fossa. The apex of the root just out into the inferior surface of the cerebellum, and you may see here. The central canal of spinal cord opens into the posterior inferior angle of rhomboid fossa, while in the anterior superior angle of the fossa communicates with the aqueduct. The lateral angles terminate as two blind lateral recesses of the fourth ventricle, recessus lateralis ventriculi coacti, covering ventrally around the inferior cerebellar pedicles. The root of the fourth ventricle, tegmen ventriculi coacti, is tent-like or fasciculum and is composed of two medullary villi, velum medullare superior, stretched between the superior cerebellar pedicles, and velum medullare inferior, a paired structure adjacent of the floccular pedicles. Part of the roof between the villi consists of the cerebellar matter. The inferior medullary velum is supplemented by a layer of the pia mater telechorioida ventriculi coacti. You may see it here. Covered on the inside by a layer of epithelium, lamina chorioida epithelialis, a rudiment of the posterior wall of the posterior cerebral vesicle. Telechorioida completely closes the cavity of the ventricles first, but in the process of development, three openings form in it. Immediate aperture, apertura megendii, this is foramen in the region of the inferior angle of rhomboid fossa, and two smaller lateral aperture megendii, and two smaller lateral apertures in the region of the lateral recesses of the ventricle, apertura lateralis lusca. By means of these apertures, the fourth ventricle communicates with the subarachnoid space of the brain, as a result of which the cerebrospinal fluid from the cerebral ventricles enters the intermeningeal spaces. Cerebrospinal fluid accumulating in the ventricles of the brain in constriction of the obliteration of these apertures in inflammation of meningitis cannot drain into the subarachnoid space, and hydrocephalus develops. So next we'll talk about A rhomboid fossa. In details, a rhomboid fossa has four sides, two superior and two inferior, according to its shape, like a rhombus. The superior sides are bounded by two superior cerebellar pedicles. You may see these pedicles here. The inferior sides are bounded by two inferior cerebellar pedicles. On the midlines of the rhombus from the superior to inferior angle stretch the sulcus medianus, which divides the rhomboid fossa into right and left halves. You may see this sulcus here and right and left halves here. On either side of the sulcus is an elevation, the eminentia medialis, you may see here, eminentia medialis, formed by an aggregation of gray matter. The eminentia medialis gradually narrows downward and is continuous with the hypoglossal triangle. You may see this triangle here, under which the nucleus of the hypoglossal nerve. You may see this nucleus in a red color. Lateral to the lower part of this triangle, this is a small triangle with gray color, and it calls vagal triangle or trigonum nervi vagi, in which the vegetative nucleus of the vagal nerve is embedded. About the eminentia medialis has as swelling the facial colliculus. You may see this colliculus here, produced by the root of the facial nerve and projection of the nucleus of abducent nerve. You may see in number 9 abducent nucleus, and you may see the fibers of facial nerve. In the region of the lateral angles on either side, the vestibular area, the nuclei of the 8th pair of nerves lie here, as I told you. Some of the fibers emerging from them pass across the rhomboid fossa from the lateral angles to the median sulcus in the form of horizontal bands, stria medullaris. You may see the stria here. These stria divide the rhomboid fossa into superior and inferior halves and corresponding to the junction of the medulla oblongata of the pons. So the gray matter of the spinal cord is continuous with the gray matter of the brain stem and partly spreads at the rhomboid fossa and on the wall of the aqueduct and partly breaks up into the nuclei of the cranial nerves or into nuclei of the bundles of the conduction tracts. So, somatic sensory nuclei embedded in the posterior horns of spinal cord acquired a lateral position in a rhomboid fossa, and you may see the sensory nuclei laterally on this picture in rhomboid fossa. So you know that vegetative nuclei located in lateral horns in spinal cord. So this, in accordance with the position of the lateral horns between the posterior and anterior horns, probably to be between the somatic sensory and somatic motor nuclei in the rhomboid fossa when the neural tube spread out. And you may see in violet color vegetative nuclei here. So, as a result, in contrast to their anterior lateral position in the spinal cord and the wall of the rhomboid fossa, the gray matter nuclei arranged in rows medially and laterally. So laterally you may see motor nuclei in red color. Somatic nuclei of the 12th to 6 pairs lie in the middle row. The vegetative nuclei of the 10th, 9th and 7th pairs in the midline row, and the somatic sensory nuclei of the 8th pair are located laterally. Projection of the cranial nerve nuclei onto the rhomboid fossa. So you may see the 12th pair, the hypoglossal nerve in red color. You may see number 18 has a single motor nucleus located in the inferior part of rhomboid fossa, deep in the hypoglossal triangle. The 11th pair, the accessory nerve, has two nuclei. Both are motors. One is located in the spinal cord. You may see in number 19 here in my pointer. Next nuclei is a caudal continuation of the nuclei of the 10th and 9th pairs of nerves, and it's called nucleus ambiguus. You may see this nucleus here in number 16. The 10th pair, the vagus nerve, has the following three nuclei. Sensory nuclei of the tractus solitarius lies next to the nucleus of hypoglossal nerve. You may see this in blue color in number 21 here. Formerly, the sensory nuclei were termed the terminal nuclei because they are the termination of the pathways formed the efferent neurons. The vegetative dorsal nucleus of the vagus nerve, nucleus dorsalis nervi vagi, you may see in violet color here, lies in the same region, trigonum nervi vagi. So motor nucleus ambiguus is common with the nucleus of the 9th pair, and it's embedded to the reticular formation deeper than the dorsal nucleus. The 9th pair, the glossopharyngeal nerve, nervus glossopharyngeus, also contains three nuclei. Sensory nucleus of the tractus solitarius lies lateral to the nucleus of the hypoglossal nerve. You may see in blue color here, it's common nucleus. The vegetative sensory nucleus is inferior salivary nucleus, nucleus salvatorius inferior in number 12. Its cells are scattered in the reticular formation of the medulla oblongata between the nucleus ambiguus and the alveolar nucleus. And motor ambiguous nucleus is common with vagus and accessory nerves. The 8th pair, the auditory nerve, nervus vestibulocochlearis, or nervus octavus, has numerous nuclei which are projected onto the lateral angles of the rhomboid fossa in the vestibular area. The nuclei are separated in two groups according to the two divisions of the nerve. One division of the nerve part cochlearis, the cochlear nerve or nerve of hearing proper, has two nuclei, dorsal one and ventral. Also it has four vestibular nuclei. You may see cochlear nuclei and vestibular nuclei. There are four nuclei. The medial vestibular, lateral vestibular, superior vestibular and inferior vestibular. The presence of four nuclei in man reflects the early stage of phylogenesis in which fish had a number of separate perceiving static apparatuses. The seventh pair, the facial nerve, facial plus intermediate nerve, has one motor nucleus of facial nerve. You may see it in number 10. Um, so the nerve fibers arising from this nucleus and form a loop deep in the tissue of the pons. This loop protrudes the rhomboid fossa as a facial calicurius, and you may see this loop in number 11. The nervus intermedius, second part of seventh pair, whose course is intimately connected with that of the facial nerve, has two nuclei. The vegetative secretory nuclei in number 12, nucleus salivatory superior, embedded in the reticular formation of the pons dorsal to the nucleus of facial nerve, and a sensory nucleus of tractus solitari, as I told you, in blue color in number 21. The sixth pair, this abducent nerve, nervus abducens, you may see number 9, has one motor nucleus embedded in the loop of the facial nerve. The fifth pair, the trigeminal nerve, has four nuclei. You may see the sensoric nuclei here, superior sensoric nucleus of the trigeminal nerve, nucleus sensoris principalis nervi trigemini, is projected on the dorsolateral part of the superior part of pons in number 29 here. The nucleus of spinal tract, you may see number 22, is a continuation of the superior sensory nucleus of the trigeminal nerve and extends for the whole length of the medulla oblongata to the cervical part of spinal cord, where it comes in contact with the gelatinous matter of the posterior horn. The motor nucleus of trigeminal nerve, you may see number 7 here in red color, lies medial to the sensory nucleus. It's concerned with mastication. The mesencephalic in number 31 here, the mesencephalic nucleus of the trigeminal nerve lies lateral to the aqueduct. It's a nucleus of proprioceptive sensation for the muscle of mastication and the muscles of the eyeball. It possibly reflects the independent development of the first division of the trigeminal nerve, the ophthalmic nerve called nervus ophthalmicus, related to the organ of vision, which explains the location of the nucleus at the mesencephalon. Let's talk about mesencephalon or midbrain. So you may see this part here and cross section here. The midbrain or mesencephalon develops in the process of phylogenesis under the predominant influence of the visual receptor, and its most important structures are therefore concerned with the innervation of the eye. The centers of hearing also form here, and together with the centers of vision developed later to form four rounded projection. You may see in number 7 quadrigeminal or tactile lamina. With the appearance of the auditory and visual analyzers in the cortex of the prosencephalon of higher animals and man, the mesencephalic auditory and visual centers themselves acquired a subordinate character and became intermediate subcortical. With the development of the prosencephalon in higher mammals and men, conducting pathways connecting the cortex of the telencephalon with the spinal cord started passing through the mesencephalon. You may see this part cerebral pedagos. As a result, the mesencephalon contains the following structure: subcortical center of vision, there are nuclei superioris or caliculae superioris, and also you may see the nuclei of third pair of cranial nerves, oculomotor nerve. So, subcortical center of vision and nuclei of nerves innervating the muscles of eye. Next, subcortical auditory sensor, nuclei which situated in inferior calicula. You may see inferior calicula here. All ascending and descending conducting pathways connecting the cerebral cortex with the spinal cord and passing through the mesencephalon. And fourth, bundles of white matter connecting the mesencephalon with the other parts of the central nervous system. In accordance with this, the mesencephalon, which in man is the smallest and structurally simplest part of the brain, has two main portions. The root or tectum mesencephaly, in which subcortical auditory and visual centers are located, and the cerebral pedicles, in which the conducting tracts mainly pass. You may see these pedicles here. The dorsal part of the root of the mesencephalon or tectal lamina is concealed under the posterior end of the corpus callosum and is subdivided by two transcending grooves, transverse and longitudinal, into four Y elevations, bodies of caliculae arranged in pairs. The two superior pairs are the subcortical visual centers. Both inferior caliculae are subcortical auditory centers. The pineal body in number 8 is lodged in the shallow sulcus between the superior caliculae, but this is part of diencephalon. Each caliculus is continuous with a brachium of the midbrain. You may see the brachium in number 11, brachium of caliculi superioris, and number 12, brachium of caliculi inferioris. So, the superior brachium passes under the thalamic pulvinar to the lateral geniculate body. You may see the lateral geniculate body in number medially than number 10. You may see under my pointer. So the inferior brachium passing along the superior margin of the trigonum lemnisci to the lateral mesencephalic sulcus sinks under the medial geniculate body, corpus geniculatum mediale. You may see the caliculus inferior and brachium caliculi inferior here, and also you may see medial geniculate body. The ventral part, the cerebral pedicles, contains all the conducting pathways stretching to the prosencephalon. The cerebral pedicles are two thick, semi-cylindrical white curds of a clearly detectable longitudinal fibrous structure with slightly spirally curved fibers. They stretch from the superior margin of the pons upward laterally, diverging at an angle of almost 80 degrees, and then sink into the tissue of the cerebral hemispheres. At the entry into the hemispheres, the optic tract crosses the pedicles. The cavity of the mesencephalon, remnant of the primary cavity of the middle cerebral vesicle is called the cerebral aqueduct, and you may see number 18 here. Aqueduct of Sylvius. Its narrow canal, 2 cm in length, lined with ependyma, and connects the fourth ventricle with third ventricle. The aqueduct is bounded dorsally by the tectal lamina and ventrally by the tegmentum of the cerebral pedicles. The internal structure of mesencephalon has following three main parts of the brain. So the tectal lamina, we talked about this part. The tegmentum, you may see this part here between tectum and pedicles, and you may see the ventral part or cerebral pedicles here. In accordance with the development of the mesencephalon under the influence of the visual receptor, it contains different nuclei related to innervation of the eye. In low vertebrates, the two superior caliculi, you may see this calicula here, in number 2. Two superior caliculae are the main site of termination of the optic nerve and are the visual center. With the transposition of the visual centers into the prosencephalon in mammals and in man, the persisting connection of the optic nerve to the superior caliculus is important only for reflexes. The fibers of the lateral lemniscus terminate in the nucleus of the inferior caliculus and the medial geniculate body. And I show you these parts here. Calicular body, medial geniculate body. The tectal lamina has two-way connection with the spinal cord through the spinotectal tract and the tectobulbar and tectospinal tracts. After decussating in the tegmentum, the tectospinal tracts pass to the muscular nuclei located in the medulla oblongata and the spinal cord. This is the visual auditory reflex tract which is mentioned above the description of the spinal cord. The tectal lamina can therefore be regarded as a reflex center for different types of movements occurring mainly due to the effect of visual and auditory stimuli. The cerebral aqueduct is surrounded by central gray matter. You may see number 19. Function related to the vegetative system. In this matter, under the ventral wall of the aqueduct in the tegmentum are embedded the nuclei of two cranial motor nerves, oculomotor, third pair on the level of the superior caliculi, and the trochlear nerve, fourth pair, but we can't see the nuclei. We only can see the trochlear nerve here. So the nucleus of the oculomotor nerve, you may see number 6, consists of several parts according to innervation of several muscles of two eyeballs. Medial and to the back of it is another small, also paired vegetative accessory nucleus accessorius. You may see number 7, or Yakubovich nucleus, and so you may see unpaired median nucleus here. So oculomotor nerve is related to the parasympathetic system. You may see to these nuclei accessories and impaired nuclei which are related to the parasympathetic system. Um, so also you may see the gray substance, gray matter between the tegmentum and peduncle, substantia nigra, black substance. Uh, you may see this nucleus with pigment melanin contained in its nerve cells. Substantia nigra extends for the whole length of the cerebral peduncle from the pons to diencephalon. In function, it's related to extrapyramidal system. The crus of cerebrum, located ventral to the substantia nigra, contains longitudinal nerve fibers which descend from the hemispheric cortex to all parts of the central nervous system located below it. The tegmentum, which is situated dorsal to the substantia nigra, contains for the most part different ascending fibers and nuclei of gray matter, the most important among which the red nucleus. You may see a red nucleus here. This elongated sausage-shaped structure extends in the tegmentum of the midbrain from the hypothalamic region to the opposite diencephalon to the inferior colliculi, where it gives rise to an important descending rubber spinal tract, tractus rubrospinalis, connecting the red nucleus with the anterior horns of the spinal cord. On emerging from the red nucleus or the rubber spinal tract decusates with the collateral tract and the ventral part of the median suture to form the ventral tegmental decussation or furrow decussation. The red nucleus is a very important coordinating center of the extrapyramidal system and is connected to all its other parts. It receives fibers from the cerebellum which pass to it in the superior cerebellar peduncles after their decussation under the tactile lamina ventral to the cerebral aqueduct, as well as from the pallidum, the most distally located and the oldest of the cerebral ganglia, which are components of the extrapyramidal system. Owing to this connection, the cerebellum and the extrapyramidal system affect the whole skeletal musculature through the red nucleus and the rubber spinal tract arising from it and induce unconscious automatic movement. In addition to distending longitudinal fibers, the tegmentum contains ascending fibers forming the midbrain continuations of the medial and lateral lemnisci. So you may see medial lemnisci here. All sensory tracts except for the visual and the olfactory ascend in these lemnisci to the cerebrum. The reticular formation, you may see a reticular formation here, and the medial longitudinal bundle are also continuous with the tegmentum of the midbrain. The medial longitudinal bundle takes its origin in different parts. One part arises from the vestibular nuclei, passes on either side of the midline directly under the gray matter of the floor of aqueduct and the fourth ventricle, and consists of the ascending and descending fibers running to the nuclei of the third, fourth, sixth, and ninth cranial nerves. The medial longitudinal bundle is an important association pathway that connects different nuclei of the nerves of the eye muscles, which causes the conjoint movements of the eyes in deviation to the side. Its function is also bondsman with movements of the eyes and head in simulation of the equilibrium apparatus. So next we talk about prosencephalon and forebrain. So the forebrain develops in association with the olfactory receptor and is at first in aquatic animals a purely olfactory part of the brain, rhinencephalon. With the change of animals from life in water to life in the atmosphere, the role of the olfactory receptor grows. With the help of this receptor, it becomes possible to perceive the presence in the air of chemical substances which make the animal aware from a far distance of prey, danger, and other vitally important natural phenomena. This is distance receptor. Um, so, other part of prosencephalon or forebrain, diencephalon, developed in a process of phylogenesis together with the mesencephalon and not from the forebrain as a result of its division. This explains the connection of the visual receptor not only with mesencephalon but also with diencephalon and its thalamus or optic thalamus. So you may see thalamus as a part of diencephalon here. So the diencephalon lies under the corpus callosum. You may see corpus callosum and diencephalon and fornix, and its sides merge with the hemisphere of the telencephalon. According to the function and development of the prosencephalon, two main parts are distinguished in the diencephalon. The dorsal, phylogenetically younger, or thalamencephalon, you may see here, is the center of efferent pathways, and ventral, phylogenetically older part, hypothalamus, the higher vegetative center. The third ventricle is the diencephalic cavity. So you may see telamencephalon here and three parts: thalamus, epithalamus, and methithalamus. The thalamus is a large paired mass of gray matter in the lateral walls of the diencephalon on either side of the third ventricle. Its edge shape with the interior and tapered to form the anterior tubercle, the posterior end expanded and thickened and forms the pulvinar. So you may see pulvinar thalami number 9, and also you may see it here in this MRI investigation. The separation into the anterior end and the pulvinar corresponds to the functional division of the thalamus into center of the efferent tracts and the center of vision, posterior end. The dorsal surface is covered with fine layer of white matter, stratum zonale. Its lateral surface faces the cavity of the lateral ventricle and is separated from the adjacent caudate nucleus by the terminal sulcus. You may see the terminal sulcus here, and you may see also the caudate nucleus of telencephalon. This sulcus is a border between diencephalon and telencephalon. A band of brain matter, the stria terminalis, passes in the sulcus. So, the thalamus is a great functional, has great functional significance. It is station for the efferent tracts. part of the fibers of the optic tract, subcortical visual center. Um, so next we talk about epithalamus. You may see epithalamus here. So it contains epiphysis. You may see number 8. Also habenuli, triangle area, trigonum of habenula. So the pineal body is related to the endocrine glands in structure and function. Bounding posteriorly into the mesencephalic region, it's lodged in the groove between the superior caliculi here of the tactile lamina, forming as it were a fifth caliculus. Dorsally, the pineal body is suspended on the habenuli, while ventrally it's joined with the medullary lamina, which carrying posteriorly is continuous with the tactile lamina. The carving of the lamina forms the posterior commissure, commissura posterior. Between this commissure and habenular commissure is small pineal recess. You may see this pineal recess here, and also you may see corpus pineale, pineal body here of this picture. So metathalamus. Metathalamus is situated behind the thalamus with two small elevation, lateral and medial geniculate bodies. The medial geniculate body, you may see under my pointer, is smaller but more pronounced. It lies in front of the inferior brachium under the thalamic pulvinar, from which it's separated by clearly defined sulcus. The fibers of the lateral lemniscus terminate in it, as consequence of which its subcortical auditory center together with the inferior caliculi. The lateral geniculate body, you may see under my pointer, is a large flat elevation of the inferior lateral surface of the pulvinar. Most of the lateral part of the optic tract ends in it. As a result, together with the pulvinar and the superior caliculi, the lateral geniculate body is a subcortical visual center. The nuclei of both geniculate bodies are connected with the cortical ends of the corresponding analyzers through the central tracts. Next, we'll talk about hypothalamus. You may see this region here. So the term hypothalamus, in the broad sense, embraces structures located ventrally under the floor of the third ventricle. And also you may see the cavity of the encephalon, third ventricle, here. It's situated in front of the posterior perforated substance, including the subthalamic region. According to the embryonic development, two parts are distinguished in the hypothalamus. The interior optic part, you may see chiasma opticus, optic chiasm, under which term are united the tuber cinereum and with the infundibulum and hypophysis cerebri. So this first term, anterior hypothalamus, included chiasma opticus and tuber cinereum with hypophysis. And posterior part is olfactory part, consisting of the mammillary bodies. You may see these bodies in red color, and the subthalamic region. Tuber cinereum located in front of the mammillary bodies, you may see here, and orange color here. This is unpaired hollow projection in the floor of the third ventricle and consists of thin plate of gray matter. The apex of the tuber is tapered to form a narrow hollow infundibulum on those blind, and the hypophysis cerebri of the pituitary gland lodged in depression of the sella turcica. The subtuberous cinereum contains nuclei of gray matter which are higher vegetative center, causing an effect in particular on metabolism and thermoregulation. The chiasma optica lies in front of the tuberous cinereum and is formed by crossing of the optic nerve. The mammalia bodies, corpora mammillaria, are two small white elevations of irregular spherical shape lying symmetrically on either side of the midline in front of the posterior perforated substance. Two gray nuclei are contained inside each of the bodies under the surface layer of white matter. The mammillary bodies are functionally related to the subcortical olfactory center. Also, you may see in red color. The subthalamic region is a small area of brain matter under the thalamus, from which it's separated by the hypothalamus sulcus here. This sulcus can be seen on median section of the brain, this one. In the subthalamic region, lateral to the substantia nigra, lies an oval body, the subthalamic Lewis nucleus, corpus subthalamix Lewisi, which belongs to the diencephalon. It's one of the links of the extrapyramidal system, and it also believed to be concerned with the vegetative functions. So let's talk about third ventricle. The third ventricle lies on the midline on the frontal section of the brain, and it has the appearance of a slit-like cavity. The lateral walls of the third ventricle are formed by the medial surfaces of the thalami, which are bridged almost in the middle by the interthalamic adhesion. You may see this adhesion here. The interior wall of the third ventricle is formed below by a fine terminal lamina and farther upward by columns of the pharynx. You may see countered of columns. On both sides, at the anterior ventricular wall, the columnar pharynxes, together with the anterior ends of the thalami, form the boundaries or interventricular foramina. You may see this foramina here, only one foramen, right foramen. Foramen of Monro, through which the third ventricle communicates with the lateral ventricle situated in the cerebral hemispheres. The superior wall, root of the third ventricle, lies under the fornix. You may see the fornix and corpus callosum here. It's actually formed of tela-chorioidea of the third ventricle, and its components are the underdeveloped wall of the cerebral vesicle remaining as an epithelial lamina and the pia mater fused with it. The chorioid plexus of the third ventricle lies on either side of the midline in the tela-chorioidea. The posterior wall of the ventricle are the cabenular and posterior cerebral commissures, between which is caudally directed blind projection of the ventricle, the pineal recess. You may see this pineal recess here. The aqueduct communicates with third ventricle here by means of funnel-like opening ventral to the posterior commissure. So the narrow inferior wall of the third ventricle is separated on the inside from the lateral walls by the hypothalamic sulci and corresponding to the posterior perforated substance mammillary bodies and the tuber cinereal with the optic chiasma on the base of the brain. The floor of the ventricles form two recesses, you may see on this picture. Infundibular recesses, recessus infundibuli, projection in the tuber cinereal and the infundibulum, and the optic recess, situated in the front of the chiasma. The inner surface of the third ventricle is lined with the epidema. So, the lecture is over. Thank you very much for your attention. Goodbye.